Welcome to Symbiota, your home to build robust genetic circuits rapidly and reliably. Today we're going to go through how to design your genetic circuit before you assemble it. So once you've created an account, you'll see your icon show up right here. You can go to your dashboard by clicking Go to Dashboard, and then you find the project that is associated with your RDP Kit purchase. If you're in a class, you may have to ask your instructor for a link or how to join, but after clicking on the appropriate project, you'll see an overview for this project, but for this exercise, you're going to need to click the Sequences tab. Within the Sequences tab, you'll see there are two libraries, an RDP parts library, which include all of the DNA sequences relevant to your kit, and a Your Circuits library, which is where you will save your sequences and your DNA designs. You can start by clicking the RDP parts library, and then you click the All checkbox, and then you open in Designer. You'll see all of the sequences open on the top, and you can click on the tabs to learn more. For example, this is the DNA code for red fluorescent protein gene. Start your design by clicking the plus sign in the top left corner of the screen. Click on the Designer tab. Enter your name, and click Create Sequence. Because the magnetic beads already come with the anchor part attached, pick and place the part into the box on the left. Go ahead and look through the parts available and design your circuit. Make sure the X's match with the X primes and the Z's match with the Z primes. The last part is always a cap, because the cap binds to the anchor and causes the DNA to circularize after elution. Now click on the book on the left and click Circularize. Because your sequence starts with an anchor and ends with a cap, you can then perhaps update your sequence name if you've changed your color, for example, and you can add a description. Click the speech bubble on the left, and you can save this to Your Circuits. If you want to see what your circuit actually looks like, click on the drop down and Edit Sequence, and you'll see the plasmid format of what your plasmid actually looks like, and the plasmid that you'll be transforming into a bacteria. Now navigate back to your Symbiota project. You can refresh that. And under Your Circuits, you will see your sequence. You can hyperlink this sequence into a lab book entry if you choose to create a lab book entry after your experiment is complete.